Okay, so first of all, I would like to thank you, the Associazione, obviously, for having me here today. I'm very excited and a little bit emotional because we are entering this field with my team like it's completely new, it's new for us, and I've been working on collagen in my doctoral study, like on the ancestral form of collagen from marine organisms. So it's really a jump, and now that we are at Ospedale Pediatrico Bambino Gesù working on rare disease, this connection is really very interesting to me. So my presentation, I will have a little bit of introduction on our lab and what we do at Ospedale Pediatrico Bambino Gesù, focusing on rare diseases. More in details, what is our aim with using zebrafish model for modeling and understanding the mechanism of rare disorders, especially focusing on brain disorders, and then the main objectives of the project, of the pilot project that was just introduced by Camerini, the Teleton-funded project, and then our ongoing efforts. So a little bit about our mission in our lab at Ospedale Pediatrico Bambino Gesù. We have a research focus on undiagnosed patients that was launched in 2015 and was one of the first study of this kind in Italy, and was focused really on sequencing undiagnosed patients and try to really help and give some sort of diagnosis there. But of course, this was not enough, and soon, so I was recruited exactly to start the zebrafish lab, because you not only need the sequencing of the patients, but you also need to combine functional studies that could be in vitro, and in my case, in our laboratory, in vivo studies. We used zebrafish model, and I will give you a little bit of introduction because I know I'm the first of two zebrafish talks, so I have the duty to introduce a little bit the model. So the idea here is that zebrafish allows for very fast and rapid functional investigation that can give you an idea on variants of unknown significance. And so this would help strategize also large animal experiments, like for example in mouse, or preclinical studies towards therapy. So we really consider zebrafish as a model to somehow speed up a little bit the functional understanding of the impact of variants that we find in our patients at the level of the organismal biology. And I think a crucial feature of zebrafish is that allow to ask a question in a global organism, because I will give you a little bit what are the features of the zebrafish, but basically they are small, transparent, you can look potentially at all the organs you are interested in without invasive technique. So this really enables you a fast functional validation, and it's meaningful because it is a vertebrate model system and has very high conservation of genetic and also cellular and organismal process during development. Okay, these are a little bit of facts and numbers about the bigger picture of the functional genomic lab and the work that was started by Marco Tartaglia at Ospedale Pediatrico Bambino Gesù, and started with this very little project in the hospital that now is a pediatric network, Rete Idea, that helps solving more than 40% of undiagnosed cases. And as you can see, more than 50 new disease genes were identified during this project from 2015 till now ongoing. And the zebrafish lab is part of this concept, and it's dedicated to a subset of this functional validation, of course. And the idea here is really, as I was telling you, to go directly, because we are within a hospital, pediatric hospital, to go directly from the undiagnosed patient sequencing in vitro data to the in vivo, and then get back to the doctors. And I think this is something we do really on the daily basis. And of course, we have also a lot of fun, try to not only provide quick answers on the information on the variants, really which impact they have at the organismal level, but also to go one step further and understand really the mechanism of pathology. And this is a little bit our team. We are few people. They're all here now because they're super excited about this new project. And I mean, it's me, Katia, Giulia, Martina, Grazia, and Valeria, and they all contribute to all the projects. They really work effortlessly on this. And I'm very happy to say that this is actually the face of the people who do the work. And we were recently also certified the zebrafish laboratory as a high standard quality in the research laboratory for the hospital standards. We have different protocols ongoing, different projects, dedicated funding, including the Teleton one that I will present you here now. And since last year, we also launched educational training that is officially recognized by the Ministry of Health. And this is very important because the use of animal model and how you use them to really achieve research is really important. So now we are offering theoretical and and practical course that will enable research in Italy to work officially. They're allowed to work with this animal model and start their own lab. Okay, so a little bit about zebrafish. I told you it's a very interesting model system because, as you can see, the development, you can see on the left, yes, the development is transparent. You can see from an egg, the whole animal is developing. You can see that it will start swimming very fast in the plate, just in the dish, and this is something that you can just look entering in the lab, in the facility, and look at the embryos that were fertilized. And you can see now the tail is developed, and the animal will quickly start moving, and this means that you can follow in less than 4 days of development, the whole development of the nervous system, and whether they are functioning and their muscle are functional. So you can do a lot of assays that are important to understand your disease really in 5 days. And on the right, you will see just an overview of all the genetic tools that are available with the community, and they go for simple RNA overexpression, for example, in case of gain of function, and this is widely used. They go for morpholino approach that you will see also in our work, where you downregulate the expression of a gene. You can go to genetic modification and be like random transgenesis, but that can answer questions of gain of function and loss of function. And you can go to targeted mutagenesis and really do patient-specific mutants. Okay, yeah, and so we added a little bit to this, and working with patient-specific, like single nucleotide changes, we also wanted to improve the genotype method. So something we do in the lab is also try to improve a little bit the methodology that is used for the model to speed up even earlier the classification of the variants. So here is just an example of our recent work where we improved the genotyping of early embryos, so we can do CRISPR-Cas within 2 days, really genotype super fast the animals and then proceed to functional study. And we have data now unpublished that show that this method that we developed, that is completely minimally invasive because it only requires really few cells from the caudal fin, enables also, I mean, the fish are not affected in any way, so they are able, we can screen them for neurofunctional studies. Okay, and here is just the overview of all the other beauties of the model. I told you they are transparent. There are many transgenic fish available. You can see, you can label your cell of interest, your district, whatever you like. In this case, you can see the vessels on the right and the vessels during the development. So you can really see angiogenesis live, and you can see brain formation and function live, as we can see later. And critical stages for the analysis normally of pediatric neurodevelopmental disease would be like around 3 days, so from 2 days to 3-4 days of development, where we follow effortlessly the development and we're trying to map and screen all the elements. And of course, you have to imagine we also stay up at night to do that because they keep developing, so we follow them closely. And so during the talk, you will see these kind of stages would be the most important stages that we look at. Okay, so in our lab, the idea is that we go to 3 levels of functional validation, and we start with the first level. Normally is a medical doctor coming to us, or geneticist, where basically there is a candidate disease gene or variant, but the function is not known, the biology is very little known, and the impact of this on the development is completely not known. So this is the first level. Usually it should be very quick and fast, and we need to provide really like some sort of clue to the clinicians. But then very often this first level go to the second level, and the second level is the real functional deal, where we try to understand, I mean, now the association between variant and disease is kind of accepted at the community level, but there is no understanding of the mechanism, and of course there is no idea of mechanism-based therapy because this is unknown. So normally we really like to go from level 1 to level 2, and this, it's normally more, I think in my experience, is more the scientists asking questions back to the clinicians to try to understand whether the prediction we make in zebrafish are valid, that they have a translational value. And I'm surprising to say that many times this is the case. So it's very satisfying working in this way. And we have a third level, and normally this is a very dedicated level. And in the lab, we dedicated to rasopathies, for example. These are diseases where the linkage between variants and disease is very well accepted. There is consensus in the community, very robust, and also at the level of the mechanism is quite known, but still there is no no therapy available. And this is usually because talking about rare and ultra-rare diseases, there are no preclinical evidence. And I think this is very important also to bring, and as you know, to this community, because we need more model and more prediction and more like confrontation of data in different models to be able to really provide preclinical material to pharmaceutical companies, but also to doctors to start clinical trial. And I think this is really the bottleneck of research and the real translation of the research. But before having a model for preclinical, we need to collect more model and confront more model in terms of mechanism and disease association. So these three levels are very important to me. Okay, so we work applying all these tools and using zebrafish model. We do different research in the lab. As I told you, we focus a lot of malformation of cortical development, Golgipathies, tubulinopathies that have also involvement of central peripheral nervous system, Rassopathies, and thanks to the launch of the Teleton Seed project with the association, since just November 2024, we also work on COL4A1-A2. And so this is what I'm going to present you next, but I want to give you a little bit of just one example of one disease that we recently published and discovered thanks to the zebrafish work. So this is a massive work that done with many colleagues at Ospedale Pediatrico Bambino Gesù, clinical doctors, cellular biologists, and developmental biologists. And the main focus was really on using the zebrafish model to try to understand what was really to understand the pathogenic mechanism behind these mutations. We are talking about gain of function mutation in a Golgi resident protein, ERF3, and so far, till this sequence patients, there was not so much information about ERF3 at all, not just associated to diseases, but associated to brain development. So this is also a new disease gene, so it's very challenging. The patients, the more severe patients, presented with microcephaly, cortical atrophy, epilepsy, and so just briefly, what we achieved in zebrafish is recapitulation of the phenotype of the patients. You can see very well the microcephaly. We also narrowed down the problem at the Golgi level. So we linked the morphological development at the level of the brain atrophy to a fragmentation, a severe fragmentation that normally you see severe fragmentation, of the Golgi that you normally see in neurodegeneration condition. And actually, this is a disease where you have progressive microcephaly and epilepsy in very severe cases. And this heat map, you can see that we tested different mutants. These are all dominant mutations, and we generated this model. You can see that there is a fair recapitulation of all the clinical features between the zebrafish and the patient. And of course, what we are doing now is once the disease was classified and published, now we are enlarging our study cohort and try to understand whether we can really assign a Golgi alterational morphotype to the type of phenotype manifestation that we see in the patient. And one thing that we really like to do is, because I told you that there are some of the patients, the most severe patients have epilepsy, we want to see whether we can set up functional assay where we can predict epileptogenic susceptibility. So what we do this in zebrafish is quite established, it's very easy, and we have our patient-specific mutant, we provide the neurostimulant that usually suppress GABAergic signaling, and then we look at them, how they behave in a dish. And you can see here, I don't know if I can point, but anyway, on the bottom, here we model our patient, and what you can see is that we look at them, how they swim in the presence of light and the neurostimulant. And you can see that the mutant embryo, the zebrafish embryo that modeled the zebrafish, the patient, they have increased response, and you can see more red in the traces, so they swim much more, and we can actually follow, I don't have the data here, but we can follow all the stages of the classical stages of epilepsy till whirlpool-like behavior. Well, they swim, they are completely erratic and swim like this. So what we are doing now in collaboration with the MBL in Germany is to try to narrow down a list of genes that is potentially implicated. It could be biomarkers for this kind of disease. And because I told you how beautiful zebrafish it is, something we can do in this model without touching the animal. So the animal is behaving completely by itself, like we don't touch it, we don't open, we don't do anything, is to look at how brain is activated. And in this movie, I think I will try to play it again. I don't know if it's playing. Yeah, you can see all neurons firing. So this is all brain activity in the neurons. And so something we started to do, also, of course, preparing for this COL4A1-A2 disease, is to try to map regions of the brain that are more active upon stimulation to try to see whether you can map that in a mutation manner. So if you can see, if we can have patterns that are specific for certain mutants. And here is just an example on the right where you can see pattern of synchronous activity within the brain, within the hindbrain. You can see active neurons here firing that we can quantify. Okay, now before I switch to the first data we have on this project, I want to say that while we were preparing the grant, we were actually working on a similar disease, and it's a new disease gene, really. We have 4 or 5 patients, and they are characterized by microcephaly, epilepsy, they have brain ventricle enlargement, and enlarged cerebellar fluid space, and really here, I mean, it's quite difficult because there is really just few patients that we know around the world that at least we try to collect it. We managed to collect so far. So the only thing we know is this is a protein that is not very well studied, but it is somehow involved in the vascular stabilization within the endothelial cells. So we made a zebrafish model, the mutation were loss of function. What we saw is that I hope you can appreciate that blocking the messenger RNA translation of this protein, then we have microcephaly in our mutant, and this was rescued by the wild type form, so it's quite specific, but not rescued from the variants of our patient. And you can see hydrocephalus, so we have enlargement of the ventricle, and more exciting what we saw, and this was really in the same time I was writing for COL4A1-A2, so this is very interesting. What we saw is that they have increased hemorrhagia. We can recapitulate the increased hemorrhagia triggered under stress. So you can see that there is a very little spontaneous hemorrhagia in this fish model, but we have it increase once we trigger with light heat shock stimulation. It's a small heat stress that we apply. Okay, now for the COL4A1-A2 syndrome, I have to do some disclaimers. So first, we started in November 2024, not 14, so it's like 2 months, and also, I mean, we basically are generating the mutants that are very patient-specific mutant, and so far this is okay. So a little bit the problem, as has been explained this morning many times, that we have many mutations, many patients, but many times these mutations are found only once, only in few families. So I think a real bottleneck here, and that's why I think zebrafish is quite important and can really work together with all the other models, is to try to provide a rapid assay where we screen the mutation in a first glance, like very quickly, and then we try to focus on some of them and provide functional evidence for all the other models and also for the clinicians. Okay, so this is just the extent of mutation that Dr. Good, of course, already described. Probably is not updated yet at this point, but I mean, the idea is that we have phenotypic variability and we have many variants, and probably for many of these variants, there are no functional study in vivo. So the idea of the project is really to, in this 12-month project, to really obtain a representative patient-specific mutants and really to try to establish a workflow, robust workflow, integrated in our common workflow to rare diseases, Ospedale Pediatrico Bambino Gesù, where we try to assess the main phenotypic traits and try to compare, give scoring and compare between different mutations. And okay, so these are a little bit all the aims for the projects, and I think in the first goal, what we aim to do is to really try to establish in our lab all the different tools and protocols that allow us to then test all the vascular defects, the eye defect, the brain phenotype defects, and the muscle defects, and then in a second phase, we will follow up with the same that we are doing for the other malformation of cortical atrophy that I show you, where we want to run RNA-seq and try to understand what are the molecular markers and whether we confirm some of the markers, of course, that come from the whether human or mouse or other model. So it was interesting. Of course, so far we are in the first step here, so producing the mutants with Filippo Del Bene at Institut de la Vision, very focused on the eye development. And what we do here is really to create, via base editing, we really mutate the base that we are interested in zebrafish and try to recapitulate the human the patient. So I don't have data to show you because, of course, the animals are being injected and they are growing, so this is very early. We just started in December to do that. But so far we try to understand what can we do with the model. So something that we started to do is, to analyze the loss of function model. So the animal treated with morpholino that downregulate the messenger RNA. This will be very important for us because in any way, together with the stable mutants, we will also overexpress messenger RNA that encode for the patient-specific mutation. So we need to understand the basis that is there. And I will tell you that all the data that I show you, we were super surprised as a team to see that we actually have very penetrant phenotype, very interesting phenotype that recapitulate a lot of the patient conditions, and this is very surprising to us because we didn't expect any phenotype, very little phenotype in the loss of function mutant. So just briefly to say that COL4A1-A2 I think COL4A1 is the one that is more conserved in zebrafish. If you look at the amino acid sequence, and it's expressed during embryogenesis, as was said before, also in zebrafish, it's quite prominent, both A1 and A2. We focus on A1 on this pilot project, and as I told you, first the idea was to downregulate the expression to then overexpress when we have ready the mutant at the transient level, and then generate this very patient-specific mutant with Filippo Del Bene in Paris that we can screen. We have all the pipeline to generate them, screen them very fast as I show you, and then map them. So we expect them to be mapping them like next month or so. Okay, so for the loss of function mutant, first of all, we look at we suppress the expression of COL4A1 in zebrafish, and we look at a very particular MTJ. So it's a junction between somite in development where the basal membrane is secreted, is formed, and then it's a very easy way to look at secretion in basal membrane. As you can see in the control up, we have expression like really a lot of protein secreted in this basal membrane, and in our deregulation model, we don't have, it's more than 60–70% of the regulation. Our model, in general, what we saw that we didn't expect, there is a dose-dependent effect on global developmental delay. They have these large ventricles that you can see in the brain. This is zebrafish brain, and okay, maybe, let's see. No, I don't know whether. You don't see it, right? No. Okay, so yeah, just follow. Okay, so you can see that there is enlargement of the ventricles. We have some hemorrhagia going on, very faint that you can see. And so the phenotype is quite penetrant, and it's also dose dependent. And if you look now at the brain, all these ventricles, behind this ventricle, we have very reduced cerebral mass. So we have a very enlargement of the ventricle, as you can see, a control and a downregulated mutant on the right. There is very little brain. And this is one of the most severe cases. I have to say that for brain malformation, this is one of the most severe cases. We have very different cases. But in terms of enlargement of the ventricle, this is something that we really have very common in our embryos. Also, the vasculature of the brain, the central arteries are completely reduced, as you can see here. So this is also a phenotype that is quite evident, and something we focus on the eye development because, of course, we know that COL4A1-A2, together with the others, but especially COL4A1-A2, are also expressed in the optic fissure, very important during the eye development. You can see it here, and in our immunohistochemistry, you can see it with the gray, it would be the COL4A1 expression, and you can see it's quite evident in the eye and the lens basal membrane, and also in the optic fissure, and this is where laminin is, so you can see we can follow the very basal membrane there of the optic fissure. And actually, in this region is very important because it's where the early vasculature in this optic fissure form in the fish, and you will have the hyaloid system formed there that then goes through 3 stages, and the branching network goes to the refinement and the maturation of the final network that you see here, right, covering the lens. And what we can see, what we can see in our mutant is also that, first of all, we have microphthalmia that is very, very important. It's something that we see. And we have, you can see the collagen expression is very dysregulated in the eye. We also have very severe cases where the lens is detaching, so it's very the tissue is very very soft and can detach quite easily in severe cases. And also we have the optic fissure failed to close in some of the embryos. It's not significant, but it's something that we can quantify and we start to see. So we definitely have an eye phenotype there. And also the hyaloid system within the back of the eye is unresolved with undefined vasculature of large caliber. This is something also we see very well. So we definitely can have a very strong eye phenotype in our mutants. And to our surprise, we can also follow in this mutant, and I have to say this is with the high dose of morpholino that downregulate. We can also see cataract, okay? So you can see on the top, you have the lens that is zoom out, and you have the development from this critical stage window that I told you, from 2 days to 3 days. You can see that the lens is very nice and smooth and uniform, and there is no development of cataract, while in the embryo, you can see below, in the mutant embryo, you can see that there is deposition of this fibrous tissue within, which is characterized very well. I think it's quite standardized in zebrafish as a cataract phenotype, and you can see this in quantification on the right, how it develops slowly by time, slowly in the time of zebrafish, like by 3 days we can have the cataract, and then this is also dose dependent on our downregulation. And then just to, for the last part, of course, we were interested also to see, okay, I mean, also in the mouse model that lack COL4A1-A2, I've seen that hemorrhage is something that occurs also, and also as Dr. Good showed us, this is also happens in his haploinsufficiency model. So we were looking at intracerebral hemorrhage. So what we did, we apply, okay. We apply, I mean, we work a little bit on the protocol. We apply a minor and major mechanical stimulation protocol on our embryo for 3 minutes. We fix the embryo, we stain them for the staining that stains the blood, and then we bleach the pigments so that you can see there are some black pigments that, of course, I mean, it's true that this transparency, the embryo is transparent if you bleach the pigment or if you have pigment mutants. In this case, we bleach the pigment afterwards, and we can see the whole brain. And what we notice is that very early during development, so around 2 days after we do our genetic modification, the controls do not show any hemorrhagia, and the knockdown show hemorrhagia. And this is also increased with the mechanical stimulation. So we reach this level where also the control, the non-injected embryos, show at least 16% of hemorrhagia. So we went back to the minor mechanical stimulation because we wanted to see whether we can set up a protocol to see really also subdural hemorrhagia events. And as you can see here, with the minor with the minor mechanical stimulation, we have many different types of intracerebral hemorrhagia. We have intraventricular hemorrhagia. We have also retinal hemorrhagia. I think I've not shown here. But this is, again, depending a little bit on the dose. And also, it's not occurring in our in the control. So it's something definitely related to the model. And it's something very striking. And again, before, these are data from early embryo. Here we go at 4 days when the blood barrier is completely formed and the animal is swimming around, so we can see that after minor stress, they have this increased hemorrhagia. I think you can also appreciate on the graph on the left that we still have hemorrhagia also in a spontaneous case. So spontaneous hemorrhage for us was not evident very early in development, but we could see it already. We could see it at 4 days. So I think I mean, once we are ready with the mutants next month, I think we have a good assay here where we can compare the impact of the mutations compared to the loss of function. Of course, go down on the dose on the loss of function and trying to see the whole spectrum from the loss of function to the gain of function. And I think to this point, I want to say that it was very interesting, the discussions we had in the morning, where also stop codon and loss of function could provide a very penetrant phenotype at the level of intracerebral hemorrhage, because I think we were very surprised by our fish data. And I mean, we do have a very 37%, I think it was the same that was shown before for the French court, if I remember. So we also have a very interesting, I mean, data set comparison here that we can talk about. And here is just a comparison stage-wise, but depending on the extension of the hemorrhage, because I told you, of course, if we push the system with the mechanical stimulation, of course, also the spontaneous, the controls will have some hemorrhage, but this in extent is very limited as compared to our mutant. So this is also well quantified here, and I think we are ready for the mutants then. So in conclusion, I think this just summarizes all the different, let's say, parameters that we have been playing around with our model so far, just waiting for the mutants. We did the loss of function mutant, but it turned out to be quite relevant for recapitulation of the disease. And you can see that we can score almost all the phenotypes that we are interested in, and we can do that in fairly little time, and we can provide support and provide pre-screening or support to the mouse model mutants. So I think this is quite interesting. Here you just see an overview on the control compared to our mutant of all the different vascular phenotype, eye phenotype, microphthalmia, and also the hemorrhage. Okay, so of course we are eager, if the conference was maybe 2 months after, I will show you already this data. So I think they are too preliminary to show at this point, but I think we are collecting interesting data also on the mutants. Some of the mutants have been selected. And something we're also trying to do, this is very preliminary, like almost like just lab data that I should not show now, but I show you because I like to share that. So of course, because we at zebrafish, we like to say that it's so fast and so easy, we also want to see secretion immediately, no? We want to test our mutant and we want to correlate and do scoring immediately with secretion. So we've been playing around with this plasmid that contains, of course, a GFP tag COL4A1, and so far I am only showing you the first data that I just scanned last week. And what we can see on the top, you can see the immunofluorescence for COL4A1 that you can see labels, as I told you at the beginning. This is our target region for basal membrane that we can look very easily, is available to us. We can immediately see it under the microscope. And below is the overexpression of the wild-type form, that is GFP tag, and I hope you can appreciate, like me, that we can see secretion. We can see staining. Major of the staining is in the basal membrane. So I hope this tool will allow us also to, if we now are making all the mutants with this GFP tag, and I hope we can also correlate the whole phenotypic spectrum that I've shown you that we are capable of with the secretion, and then come to the next conference with an overview of all the data set and compare it with the other models available. And with this, I thank all the members of Marco Tartaglia's lab, of course. Especially, I thank the zebrafish team that work on this project, all the fundings and the collaborator, and Filippo in Institut de la Vision, who is very interesting in working with me on the eye phenotype and generating the stable mutants. And thank you for the attention.
Thank you, Antonella. It was really exciting to listen to so many informations, and really, great work. In a few months, so many information. So
the model. I think I'm selling the model.
You start from the model. So, any question?
Congratulations for such an amount of data in a so short period of time. It's wonderful. I have a question. Do you have bleeding with or without mechanical stimulation in other areas, not the brain and the eye? That's my first question. Yeah.
I answer the first? Okay.
Yes.
So thank you. This is really the work of the whole team who's working like crazy on this project that we really like. And yes, we do hemorrhage. We do have hemorrhage in other sites. I'm not quite sure how to describe them yet. So we focus on intracerebral, but I have to say in the spontaneous, I think, and they might correct me if I'm wrong, because it's data from the people of my team, but I think we do have hemorrhage in the posterior region of the embryo, not in the brain. But I wouldn't know now exactly which vessels the leakage is from, which kind of system. But yes, we do observe it, I think, in 15 to 16% of the cases.
Because you mentioned aberrant trunk vessels?
Yeah, there are malformations also in the trunk vessels that for...
Which kind?
Okay, we see overall reduction of the trunk vessels morphogenesis, and but blood flow is fine in the low to average dose of the knockdown, and I think it was in one of the graphs I showed there is a certain percentage in the trunk vessels of leakage, yes. But we haven't done the study. I mean, this is very, very preliminary, so I can tell you yes, but I cannot say, I cannot go in details of that. I would say something that is not correct.
Any other question?
I mean, I don't know if it makes sense, but more in the spontaneous than in the mechanical trauma.
Because patients never bleed.
Yeah.
I mean, as far as I have in mind, patients never bleed outside of the brain. And
That we are aware of?
This is, yeah, that has been published or seen, or we have this hemorrhage, and we have these aneurysms also. They might have, for sure, in some patients, they have aneurysms elsewhere.
Elsewhere?
In splenic artery, mesenteric artery.
Maybe
But I just wanted to know.
we could compare that. I don't have the
see
data to compare.
Aneurysms or?
But maybe also the zebrafish. I mean, zebrafish is very different, right? So it's swimming. The pressure is used to is quite different in early development. You have to imagine. I mean, it's external development. So I think a lot of thoughts went into how do we really apply mechanical stress that is reasonable, comparable to, to human pressure. So I'm not sure about this trunk. But definitely, it might be a very specific zebrafish thing that we will see when we will have all the data and maybe talking with you and trying to see whether there are other sites and we can compare them anatomically and evolutionary, That I'm not aware. At this point, I wouldn't comment on it.
The last question, if I can. Have you planned to do not mechanical stimulation and have your little fish growing, becoming adults, and then see what's the phenotype? And so this is ongoing also, I guess.
This is ongoing. I mean, they will have 1 month, so we will have to wait 3-4 months. So it's before time. So, but definitely we will look at that. And I mean, we are doing that in transparent embryos. So we really see without staining, whether we have massive or an interesting amount of hemorrhage and wear.
Thank you.
Thank you.
Thank you. I have a short question, maybe it's a naive question, but when you have a disease like we learned this morning that it's there is a lot of variants, clinical variants, does it affect in the animal models, or, it's something that there is there a problem if there is a clinical variance? Because there are some patients that their onset is an adult age, and some others that they are in the embryo phase, so.
Yeah, I mean, this is a very good question. So I think this is part of the strategy. So we do have meeting to try to select, of course, the best suitable, right, candidate and variants, and that we can model. Not everything can be modeled in fish. I mean, they don't have a cortex. So if we're talking about layering, for example, I try to avoid it, but the basic mechanisms, for example, of cortex formation are conserved, so that we can answer. And in terms of more the variants, but also I think with the ARF3 study, we showed that the variability that you see in the patients, you can also recapitulate in zebrafish. And something I have to stress is that zebrafish is very polymorphic. So we have different strains, and very few strains, I mean, I think 1 or 2 strains that we have are quite homogeneous in genetics, so we like to compare also the results we get to different offspring of polymorphic strains, and I think this in its own way, in the zebrafish way, but is closer to what happens to humans, of course. So I think that this penetrance issue, I think once it's investigated and plotted, is also like a super interesting data for the community.